This is our final video for the muscular system. Next up, we'll do a little bit on the skeletal system. So we're going to talk now about cardiac muscle and about skeletal muscle tissue. One thing that I am not going to do is go into the physiology of cardiac muscle. That's somewhat complicated, and it's something that you'll cover in A&P2. But I did want to note some of the similarities between cardiac muscle and skeletal muscle. So cardiac muscle also has actin and myosin. There are also Z discs. It also uses calcium in contraction. And so basically it uses the exact same crossbridge cycle that you see in skeletal muscle. So I told my A&P2 class as they were dissecting their hearts, I said that cardiac muscle has exactly the same proteins arranged in exactly the same fashion. Therefore, it probably pretty much tastes just like um, skeletal muscle. So if somebody offers you beef heart, it's going to be basically like a pot roast. And let's be honest, if you're in coronavirus quarantine, you better take what you can get in terms of meat. Okay, cardiac muscle is involuntary. It has its own unique conduction system, so the heart will beat independently of any signals from the nervous system, which definitely makes it different from skeletal muscle. It also has autonomic innervation, which means it has portions of the sympathetic and parasympathetic system having nerve fibers attached to it. That's something that we can talk about in a little bit more detail a little later on in the semester. So basically, it means that you've got fight or flight signals that go to the heart. Okay, as far as smooth muscle goes, the first thing that I want to remind you of is where smooth muscle is located. It's located in virtually every tubular organ in the human body. So you've got smooth muscle in the walls of the GI tract. You've got smooth muscle in the tubes of the respiratory tract. You've got smooth muscle in the tubes that are in the urinary and reproductive systems. And you've also got smooth muscle in the walls of most blood vessels. And so there's a lot of smooth muscle around the body. So let's talk about it. And one thing to say is that smooth muscle contractions start more slowly and last longer than skeletal and cardiac muscle contractions. There's basically a different mechanism for contraction here. Additionally, smooth muscle can shorten and stretch to a greater extent. And this doesn't have as much to do with the way that it contracts as it has to do with the arrangement of actin and myosin on it. And we'll get into that in just a minute as well. So smooth muscle is involuntary. It's innervated by what are called the intrinsic plexuses of nerves. We'll learn more about that when we talk about the nervous system. It also has autonomic innervation, so sympathetic and parasympathetic fibers leading to it. It means that basically you're going to see different reactions during times of high stress and during times of low stress. Smooth muscle is not striated, meaning that there's no thick filaments. I'm sorry, they do have thick and thin filaments, but they're not arranged into sarcomeres. And so you don't have the same sort of I bands and A bands that you have in skeletal and cardiac muscle. And so this lack of striation is what actually gave it the name smooth muscle in the first place. So each of the smooth muscle fibers is thicker in the middle and it's tapered on the ends. There is one nucleus per smooth muscle cell. So this is in contrast to skeletal muscle cells, which have multiple nuclei, and cardiac muscle cells, which can be binucleate or have two nuclei. Some smooth muscle does have gap junctions to allow the smooth muscle cells to act in unison. But the biggest point of difference in terms of the arrangement of the actin and myosin is that there are thick and thin filaments, but they're contained in intermediate filaments, not in sarcomeres. So basically you have, if you look at the diagram, you'll see the intermediate filaments, which are sort of this cross webbing that's across the skeletal muscle cell. And you've also got dense bodies, which you can see are at the intersection of the intermediate filaments. And so the dense bodies, in terms of their function, act very much like Z discs. So they're anchor points for these contractile proteins. Now, when myosin moves actin towards itself, it's going to tug on these dense bodies. Because of the arrangement of the dense bodies over the smooth muscle fiber, you're going to see kind of a corkscrew motion as the muscle fiber decreases in length. And what I want you to think of is wringing out a dish towel. So you sort of twist it and it shortens as it goes. And so that's kind of the motion of smooth muscle tissue. You get this corkscrew motion, and that corkscrew motion actually allows the smooth muscle to shorten to a greater extent than skeletal or cardiac muscle actually can. Now we're going to take a look at the contraction mechanism. This is something that is very different from skeletal or cardiac muscle. So in this, you do have calcium coming into the cell, but it doesn't bind to troponin. What it binds to is a protein that is called calmodulin. And so when the calcium binds to the calmodulin, as was with any other protein, it changes shape. So it changes shape to an activated form. And what that activated form will do is itself change inactive myosin light chain kinases to activated myosin light chain kinases. So right here I want to remind you that kinases are enzymes that add phosphorus to another molecule. And so you've got these kinases activated, and what they're going to do is to phosphorylate other proteins. And in this case, the protein that they phosphorylate is actually myosin. Now, one thing that I was careful to remind you of when we talked about the cross-bridge cycle was that myosin has to have phosphorus attached to it in order to be able to form a cross bridge. So up to this point, the myosin molecule without a phosphorus can't form a cross bridge. And so when these kinase enzymes phosphorylate the myosin, it will actually activate the myosin. So in this picture here, I want you to note that you've got, this says inactive myosin molecule. You've actually got two of them here with two heads, and each of those myosin heads have now been phosphorylated and therefore are now active. They're ready for business. Now, something that I want you to notice about the thin filaments here, which have actin on them, is that those binding sites are already open. As it turns out, there's no troponin and tropomyosin complex associated with the thin filaments here. So the thin filaments, not having tropomyosin cover them, are always ready to go. They're always open for business. And so with smooth muscle, what you have to do is you have to activate the myosin before cross-bridging can actually occur. Now, once the cross-bridging actually occurs, you do go through the same sort of cross-bridging cycle where you get the attachment of myosin to the actin, you get a power stroke, and so forth and so on. Now, it does take ATP to break this cross-bridge, as it does in skeletal and cardiac muscle. We'll examine the implications of that in just a minute. So now what we're going to do is to directly contrast the mechanism of smooth muscle contraction with the mechanism of skeletal muscle contraction. And we're going to start out with the similarities. So in smooth muscle, you have an increase in cytosolic calcium by release of calcium from the sarcoplasmic reticulum. Likewise, in skeletal muscle, you also see release of calcium from the sarcoplasmic reticulum, resulting in a rise of intracellular or cytosolic calcium. Now, the mechanism by which the sarcoplasmic reticulum is prompted to release this calcium does differ between skeletal muscle and smooth muscle, but those differences are a little bit complex to get into in this course. The biochemistry is very different. So once you've got calcium present in the intracellular space in the cytosol, very different things are going to happen. So in smooth muscle, the calcium is going to bind to calmodulin in the cytosol. In skeletal muscle, the calcium is going to bind to troponin on the thin filaments, and from here, this is where things get very different. In smooth muscle, once the calcium has bound to the calmodulin, that calcium-calmodulin complex will bind to myosin light chain kinases. After this, the myosin light chain kinases will use ATP to phosphorylate myosin cross bridges. Once the myosin has been phosphorylated, it is able to form cross bridges with actin filaments, and once this can happen, the cross bridge cycle produces tension and shortening. In skeletal muscle, when calcium binds to troponin on the thin filaments, the troponin is going to change shape. And what this will do is to cause tropomyosin to move out of its blocking position, blocking actin from cross-bridging. So once the tropomyosin has been cleared from the actin, then the myosin can cross-bridge to the actin, and your cross-bridge cycle can produce tension and shortening. So this is where I want to draw the big contrast between smooth and skeletal muscle. So in smooth muscle, the myosin has to be activated. If you'll recall, when I talked about the cross-bridge cycle in skeletal muscle, I said that myosin has to have a phosphorus attached to it in order to be able to form cross bridges. So in smooth muscle, when the muscle is relaxed, phosphorus is not attached to the myosin, and therefore cross bridging can't occur. So basically in smooth muscle, the myosin has to be activated. The actin is always ready to go, but the myosin has to be activated. In skeletal muscle, the myosin is constantly phosphorylated by binding to ATP and then hydrolyzing it. In the meantime, when the muscle is relaxed, you have tropomyosin covering the actin. So in this case, the myosin is actually ready for business all the time, and the actin has to be activated by moving the tropomyosin out of the blocking position. So basically in smooth muscle, actin ready to go, myosin has to be activated. In skeletal muscle, myosin ready to go, actin has to be activated. And that's kind of the biggest difference in terms of the contraction mechanism. Now there's one more thing to add. When we were talking about different types of skeletal muscle fibers, I talked about the differences in myosin between these different fibers. That the rate of the ATPase mechanism made a difference in whether they were fast or slow twitching muscle fibers. Now in this case in smooth muscle, you'll notice that it takes the myosin light chain kinases to phosphorylate the myosin cross bridges. And what this means is that the myosin doesn't carry out this ATP hydrolysis on its own. It has to have some assistance in order to do that. Now, there is another thing that we're going to take a look at with smooth muscle contraction mechanism, and this is something that is called latch state. So this occurs when you're constantly sending stimulatory messages to smooth muscle, and therefore the calcium level within that smooth muscle stays elevated. Now what happens during these conditions is that the rate of ATP splitting will actually decline. So the rate of ATP splitting means that this declines, yet isometric tension is maintained. And so isometric tension meaning that there's still tension, but the length of the muscle is not changing. So what's happening here is that the phosphorylated myosin, it becomes dephosphorylated while attached to actin, and if you'll recall, that strengthens cross bridges. And as a result, this causes myosin to release from actin far more slowly. So what this means is that the cell can maintain tension without moving because these cross bridges actually stay connected. And what this results in is the ability of smooth muscle to maintain tension for long periods with low ATP use. So if you think about this, smooth muscle in blood vessel walls has to maintain tension against persistent pressure of blood flow from the heart. And so it becomes useful for this muscle to be able to maintain a state of tension, maintain a state of contraction for a long period of time. Additionally, you see these long, slow contractions in peristalsis when food is moving through the GI tract. So smooth muscle has this ability to go into latch state and maintain a contraction for longer periods of time. Ironically enough, this maintained contraction actually results from less ATP use. So this is something that I actually want to connect with something else that you already know about. So You've probably all heard of rigor mortis, which is when a corpse stiffens in the same posture that it had when the individual died. And so what is it that actually causes rigor mortis? Well, here with smooth muscle, this is an example of how ATP breaks cross bridges. And so in the absence or with low ATP, smooth muscle maintains a contraction. When you're dead, you don't produce any more ATP. So in the case of rigor mortis, this is when the skeletal muscles stay locked into the same position because there's not ATP available there to break the cross bridges. So low ATP levels or lack of ATP can result in extended cross bridging. And in the case of skeletal muscle, definitely something that you don't want. But in the case of smooth muscle, it is something that you can want to have happen from time to time under specific circumstances.